Welcome, everyone, to another episode of the Retina Channel podcast. Today, I'm very pleased to welcome back Dr. Brian Ballios from the University of Toronto and Toronto Western Hospital for an update on inherited retinal diseases. This is actually a special episode for me because Brian was the second guest ever to be featured on the Retina Channel all the way back to 2017, shortly after we started the podcast. Now, after 9 years, he's back for episode 98. So it's really nice to have him back and also see how things have evolved in the field of retinal diseases. Brian is an assistant professor of ophthalmology at the University of Toronto and also holds the J.R. Death Hill Fighting Blindness Canada Professorship in Ocular Genetics. He is a clinician scientist at Donald K. Johnson Eye Institute and the University of Toronto and University Health Network. He has clinical research and also focuses on inherited retinal diseases, ocular genetics, and also regenerative medicine. Brian completed his MD and PhD and also ophthalmology residency program at the University of Toronto, subsequently completed a fellowship training in medical retina and inherited retinal diseases at Harvard University and Mass Eye and Ear in Boston. His research spans stem cell biology, retinal regeneration, gene therapy, and other emerging treatments for inherited retinal diseases. And that's really what we're talking about today. We cover where things stand with gene therapy almost a decade into the Luxturna era, including RPE65, newer approaches such as antisense oligonucleotides and optogenetics, emerging treatments for Stargardt disease, and also evolving role of stem cells and photoreceptor replacement therapies. We also talk about some of the challenges facing the field, including clinical trial endpoints, surgical delivery of gene therapies, and also importance of good natural history data as more of these treatments become clinically available. It was a great discussion with Brian and terrific overview of where the field stands in the inherited diseases. And I'm sure you'll learn from this episode as I did from my conversation with Brian. And then, without further ado, this is Dr. Ballios.
Dr. Ballios,
Nice to see you.
nice to see you, Brian. How you doing?
I'm fine. How are you?
I'm good. Welcome back to the Retina Channel podcast.
Yeah, thank you. Thank you. I'm still at work, as you can tell.
I can tell you're in the office still. I see you're still in the office. Well,
Yeah.
it's been many years since you were here. I think you just started your practice the first time, or maybe
Yeah.
you're wrapping up your training. I'm not quite sure, but welcome back.
Thank you. Thank you. Let me see if I can get my. You're seeing my video okay? I'm not seeing it so well, but you
I
can
am
still
seeing
see.
you. I'm seeing
Okay.
your video. Can you see me?
Yes, I can see you. Yeah, that's fine.
Perfect. Perfect. Okay. Brian, I think you're the perfect person for this topic. You're leading your research on this and you're up to date with IRD and what's going on. So I thought we need an update on the Retina channel about IRD and what's going on, considering,
Sure.
so many patients we have, so many questions we get, people ask all the time, is there any hope for me? What's the status of stem cell or of implants, those things. So, and a lot of us, even those practicing retina, it's hard to keep up to speed with what's going on and, to give patients something to look forward to. So I was hoping you can bring us up to speed on a large level, a kind of a gestalt over the landscape of research and what's up and coming, what's actually materialized, what's been done on humans, even small studies
Yeah.
when it comes to things like RP or things like that. Okay. So thank you for coming.
Yeah, no, my pleasure. I think if we'd had this conversation, we'd gone back maybe 6, 7, 8 years. I think the biggest thing to talk about would be gene therapy. I think we'd be
Mm-hmm.
talking about, we'd probably talk for half an hour about the Veretagene Naparvovec, what's marketed as Luxturna for RP65 disease. It was
Mm-hmm.
new on the market. It's a very exciting time, and it still is a really incredible therapy. We've learned so much about that. I think one of the biggest things we weren't really sure of when that drug was first released was what happens to these patients when you treat them, like 5, 6, 7, 8 years later, or a decade later. We
Mm-hmm.
clinical trials don't last that long. We don't really get information from that,
Mm-hmm.
and real world studies in inherited retinal disease is just not something that the field really understands, right? We know about real world studies for anti-VEGF agents and other things, but we don't know about what that means to do in gene therapy. I think we've learned a lot about what happens to these patients years after they've been dosed, because it's supposed to be a one-time therapy, gene therapy. At least that's how gene therapy has originally been designed. Is it durable? Is it giving lasting effects? Are there adverse effects? And we've learned that in some cases there is continued progression or changes within the retina that happen even though the patients experienced a dramatic benefit or improvement in their vision. That there are parts of the retina that continue to experience degeneration. The parts that are helped and by the therapy, the function is stronger, and you can demonstrate that by visual fields or microperimetry or other types of functional, psychophysical outcomes. But you can still see anatomic progression of degeneration and disease. And
interesting.
We're seeing that over now. We're looking at 6, 7, 8 years. So what does that look like, in 20 years or 25 years for these patients? Will they need to be redosed at some point in their lives? There are still clinical trials going on for new RPE65 gene therapies. You might say that's crazy, right? Like, why would you've got a drug that was kind of a blockbuster and just changed the whole landscape.
I guess that's anti-VEGF medications. Different companies target the same thing.
Yeah. So like I said, 7, 8 years ago, we would have been talking about Luxturna and RP65, but we could still talk about it. But a lot of the focus in the last 5, 6 years has been what's happening to all of these patients that we treated with it, because we treated a lot. In Canada, across the world, there's a large study going on in Europe called the Perceive study. That's being led by Dominic Fisher. It's a terrific study, and they're collecting data now. I think they're in maybe year 4 at least of their real world study
of Luxturna patients.
Luxturna patients with RP65 with confirmed genotypes, following them out over time since their treatment that they had the one dose. So
interesting. So let's talk a little bit more about the gene therapy while you're at it. So you said right now the focus is what happens in the long run. But
that's
what other gene therapy approaches are underway or have been approved or are close to being approved? For example, ABCA4, or many other RP genes. Why does RP65 has been the target? Any other gene in the horizon to be targeted?
So one of the exciting targets in the last few years has been X-linked RP and the RPGR gene. And there was a lot of excitement around 2 large studies, and there still is, but around the data that coming out of 2 large studies. One that was a trial initiated by a company called Mira GTX for a gene replacement therapy for RPGR. They ultimately were acquired under the umbrella of Janssen, and then Johnson & Johnson. And then a second study by a company called Beacon Therapeutics. Beacon, formerly their asset was under a company called AGTC, and this work is about gene replacements. It uses the similar principle, delivering the gene in a adeno-associated virus to try to improve function, and 2 companies doing pivotal phase 3 studies. J&J released their data last year in 2025 for their pivotal phase 3. And their secondary outcomes were really exciting. Improvements in low luminance visual acuity, improvements in FST in their treated group compared to their control arm. But they didn't pass statistics on their primary outcome, which was the visual mobility course. And that still is a primary outcome for most of these studies in phase 3. It's, again, the multiluminance mobility course. It's got a number of different names now, but it essentially takes patients through navigating an obstacle course under various levels of illuminance. That's an accepted primary outcome by organizations like the FDA. They didn't meet the statistics for it. So their compound is not going to progress to a something that's going to be marketed for these patients.
Gotcha. And that's X-linked RP. That was X-linked
for X-linked RP. Beacon has not released their phase 3 data yet, They were talking about maybe releasing the second half of this year. I haven't seen the data from that yet, but everyone is waiting with great anticipation to see it. I think that there's good signal there within these studies, and I think
Gotcha.
if you look at the J&J data, you see that there's good signal there to support the rationale for doing this. But just like other studies have not always met their primary endpoints, gene therapy doesn't always meet its primary endpoints either.
And these are subretinal vitrectomy delivery systems, or are they
subretinal
injections,
vitrectomy done with raising a bleb, now usually multiple blebs in the peripheral macula, trying to sandwich the fovea. Most studies have shown that you want to get away from foveal detachment in any of these surgeries, because the fovea in these IRD patients is very sensitive to detachment. They'll tend to atrophy after it's reattached.
Already thin, yeah. Gotcha. And then are these mainly targeting kids and juveniles, and or are they targeting people in their 30s and 40s?
There's some literature being published now around not about the best populations to go after. This will lead me into one of the other things I wanted to mention to you is most of these studies in the early phases have selected people who are adults to start with lower visual acuity. And as the phases progress to phase 2 and phase 3, have moved into, if not young adults, then adolescent populations.
Yeah.
It's a little different than what was done for RP65, where in the phase 3 there were a lot of young people, adolescents and pediatrics, but that's because that phenotype was presenting as LCA. Often these patients had very challenged vision from a very young age in most of the patients with RP65 disease. But in X-linked retinitis pigmentosa, or in USH2A retinitis pigmentosa, which is also subject of gene therapy studies, these patients are presenting as adolescents. They're experiencing a lot of their loss of visual function as young adults in their 20s and 30s. And so we're getting a lot of those. But we're still seeing pediatric, quote-unquote, pediatric cohorts, or patients under 18 in the phase 3 studies in some arms. And I think we'll continue to see it. But yes, gene therapy for X-linked RP. But it's not just the traditional approaches that we're seeing in the last few years. We're also seeing work on other types of gene therapy. So RNA modifying therapies, for example, antisense oligonucleotide therapy. There's been a lot of studies and trials looking at trying to use this for USH2A, for example, which is the gene associated with recessive retinitis pigmentosa. Mutations in USH2A are the most common cause of recessive RP and Usher syndrome, but also just non-syndromic RP, and also a CEP290, CEP290 as a cause of LCA or early onset RP.
And these are mRNA or RNA injection methods.
The antisense oligonucleotide can have a variety of different mechanisms of action, but one of its mechanisms can be to skip problematic exons. So I'll give you a good example. In USH2A, there's a mutational hotspot in the gene in exon 13. A large number of mutations, clinical variants that are pathogenic, occur in exon 13. And it turns out from studies that exon 13 is dispensable for protein function. But the problem is silencing exon 13. So what antisense oligonucleotides do is they get into the cells, they're taken up from the extracellular material through this process called gymnosis, which is still somewhat of an opaque process. We don't really know the mechanism of how they're taken up from the extracellular environment. These antisense oligonucleotides are short oligonucleotide sequences. They go in and bind to the mRNA at specific areas they're designed to bind. And for example, in the USH2A around exon 13, they'll bind and they'll allow during translation to protein for the ribosomic machinery to skip exon 13. It creates a truncated form of USH2A, but still a functional protein, avoiding the problematic part of the transcript.
Interesting. So how is it repeated? How is it propagated inside the cells? Like, is it?
So it's not like so in Luxturna, the virus infects the cells with the plasmid, and then it continues to produce the protein over time. But this has to be redosed. So
Oh yeah,
it gets you
that's
back to the
go
paradigm we're all very familiar with as retina specialists:
yeah.
repeated intravitreal injections,
Injections, yeah,
right?
yeah,
So
yeah.
that's the delivery paradigm, and that's how the trials are designed. So
I see.
there's an ongoing phase 2b study on that called Luna, which is studying that for USH2A. There's been a study in called Hyperion, which is studying in CEP290 as well. It's very interesting
Interesting.
phase 2
The
studies.
antisense are antisense oligonucleotides
Yeah.
targeting mRNA, and
So
these are
it's
repeated intravitreal injections. Yeah,
exactly repeated intravitreal
yeah.
injections because the antisense oligonucleotide degrades over time. The intracellular enzymatic machinery digests it, and it needs to be
Hmm.
repeatedly dosed. So
Interesting.
intervention in the traditional, or what we think of as traditional, it's hard to say traditional, but traditional uh, gene replacement therapy, which is what you think of
Hmm.
as the RP65. People are still working on that.
Hmm.
We gave example in X-linked RP, antisense oligonucleotide, and then a whole area called optogenetic therapy, which is gaining a lot of traction, where groups are studying how to introduce light-sensitive opsins into non-photoreceptor cells in the retina, bipolar cells or
Mm.
ganglion cells, to make them become light sensitive,
Yeah.
provide them with opsin protein so that they might make use of chromophore to become activated in response to light,
Mm.
to change their membrane potential in response to light. And
Hmm.
it's a kind of a gene therapy, but the target is not photoreceptors. It acknowledges that in advanced disease the
Hmm.
photoreceptors are
They're
eliminated,
gone. The
that the inner
bipolar cells
retina,
can pick up the
yeah,
function here or
if you
something.
don't have the substrate for vision to
Hmm.
rescue with gene therapy, then you need to find a new substrate. And if there are inner retinal cells that remain, maybe if you make them light sensitive. This was some of the
Fascinating.
ideas around retinal prostheses. Remember the
Yeah.
implants and what was tried with Argus, where they implant them on the surface, so the epiretinal implants, they stimulate the inner retinal cells.
Same
It's a
idea.
similar thought, but it's using a gene therapy approach. Again, intravitreal injection, maybe one,
Intravitreal
maybe
injections.
multiple
Okay.
times. Yeah,
Okay. So I guess adeno-associated virus is introducing the
the
opsons or
compounds.
okay. Okay.
And then but people are also starting to take what we're learning. So there's been so much talk lately in comprehensive retina around GA drugs, right? Slowing
Hmm.
down the progress of GA, and we know GA is not just a disease that affects AMD. GA is present in many of our Stargardt disease patients, and
Yeah.
so really exciting work from the recent Dragon phase 3 study for a compound that slows down the progress of geographic atrophy.
Is it true complement inhibition or
It's
different
not
mechanism?
complement inhibition. It's a chemically modified form of vitamin A that patients take orally, and so it's not
Oh,
done as a local injection. It's an oral supplement of vitamin A
okay.
that's chemically modified, and it prevents
Okay.
dimerization of the vitamin A metabolite within RPE cells, which is a pathologic process involved in RP cell degeneration in Stargardt disease, or ABCA4 disease,
Okay.
I should say, in general. And this study, Dragon, which was a phase 3 study of this compound run by a company, Sepal Bio, I believe the name. I'm name dropping a lot of companies. I'm not endorsing any particular group. Just so the podcast audience knows we're just talking about some of the most recent
No,
stuff
I'm
that's
I'm
in
impressed
trials.
by your memory of these
Yeah,
different
patients come and
companies.
ask these questions all the time. They've seen it on the internet, right? Doctor
Yeah.
Google, like Google AI or whatever, can give them an answer in 2 seconds, or Chat
Mhm.
GPT, whatever. So that Dragon phase 3 study just released its primary its top line data, and showed,
Mm,
and they were approved by the FDA as their primary endpoint, GA lesion size.
interesting.
Just the way the studies like Oaks and Derby and Gather 1, Gather 2 for the complement inhibitors that are currently available right now in the market,
It's an
ACP
oral
and
medication.
pegcetacoplan,
Okay.
they were approved based on anatomic change. So too
Mm,
has this molecule been approved, or
interesting.
sorry, apologies, has not yet been approved, but their top line data shows statistically
It could
significant
be approved.
improvement in
Gotcha.
lesion size. They're filing, or I believe, I expect they'll shortly file with the FDA for approval, if not before the end of the calendar year.
Great. So just to wrap up the gene therapy approach, you had similar things, introducing the actual gene like
Yeah.
RP65. Other candidates are there. You had the antisense oligonucleotide, which very cleverly targeting mRNA molecules and blocking that, and also optogenetic modification was another approach. So there's some promising ways of maybe introducing some functioning products into the
Yeah, a lot of
cells.
these are in phase
Yeah,
2 or phase 3 studies right now.
yeah.
Big
And
studies.
different targeted genes, right? You mentioned, USHR2.
Yeah.
You mentioned RP65, BCA4. Those are also that's fantastic. So
Prevalent disorders that are like really out there.
yeah,
Yeah.
and that would be nice because RP65, I mean LCA is definitely a real disease for kids, but when we see an adult patient with RP, usually it's not RP65, right?
No.
It's USH2,
Yeah.
or something like that. So it would be good
Yeah.
for more older patients. So let's switch gears to maybe non-gene therapy. Talk about stem cells.
Sure.
That's something I get a lot of questions about. What is stem cells? What's going on with stem cells? I think it's been in the topic for decades now, but
So long.
few things have actually come out of it. So is there any hope or any progress in stem cell? We talked about this before. Stem cells are RPE stem cells, photoreceptor stem cells. Has anything been close to being done, or what's on the horizon with that regard?
Yeah, I think it's still a very active area of research. I think we've learned a lot from clinical trials and stem cell therapies. Most of the stem cell therapies, again, if we were talking, 6-7 years ago, many of them were focused on transplanting the stem cells themselves into the retina with the expectation or the hope that they would regenerate photoreceptors, that they would replace cells. I think what the field has learned a lot is that stem cells on their own don't always know exactly what to do when you put them in the eye. They may be very useful at releasing factors that support the health and survival of cells, but what people are really interested in the field or hopeful for is to replace light-sensitive cells or the RP cells to support them. And transplanting just the stem cells into the retina, into an adult retina at least, or a mature retina, they haven't really shown that they know exactly what to do. So what the newer approaches that are being explored now are focused on transplanting cells that were derived from stem cells but have already been pushed down the pathway towards differentiation into a specific cell type. So a great example is photoreceptor precursors or progenitors that are made from stem cells in the lab. The stem cells are grown at scale in large bioreactors, and you make billions and billions of photoreceptors from these stem cells. And of course, the stem cells, the advantage here is that they're stem cells, so they're immortal. They continue to grow in the lab for months and years, and as you scale them up, they grow in large quantities, and they kick off progenitors that you can differentiate into photoreceptors. You transplant those immature photoreceptors, and there is some preclinical evidence to support their ability to integrate into retina, and it's being explored now in a pretty important phase 1 study sponsored by a group called Blue Rock Therapeutics, which is a subsidiary, an independently operated but wholly owned subsidiary of Bayer Pharmaceuticals, and they're running a phase 1 study, and they're using a bank of stem cells to create photoreceptor precursors, and the hope is that after they transplant them, that they would be able to integrate into retina. And they're going after RP, and they're going after cone rod dystrophies, and the hope again with stem cell therapy is that it would also be agnostic to the genotype. So it would be gene independent, like that you wouldn't care what gene was causing degeneration. You really just want to replace the cells. There's all sorts
for macular degeneration too,
right? And people are looking at that for macular degeneration. They're thinking about transplantation of microglia in macular degeneration. I've seen that. So not just photoreceptors. People are thinking about other types of cells for transplantation. Microglia are a population of immune cells in the retina that can support the health and function of endogenous retinal cells, not just help the retina fight off infection or other things, inflammation, but they also can provide growth factor support for other cells around them. And so people have been talking about transplantation of microglia, and there are still groups pursuing the RPE transplantation for geographic disorders, not just AMD, but also in IRD, the things that come often from ABCA4 disease.
So what's the roadblock here? Is you mentioned they can somehow lead those stem cells to progenital cells for photoreceptors. So it sounds like we know we do have technology to sway them that way. Is the roadblock getting them to stick in the eye, not getting rejected, or continue to become full photoreceptors, or what's the hindrance here?
I mean, a lot of hypotheses. One of my hypotheses is that it's a lot to do with integration of cells and how they connect, and it's something we explore a lot in our own research and in my lab, and a lot of groups around the world are exploring how to improve the ability of cells to integrate. There has been a lot of very, very interesting preclinical work that's focused primarily on how do transplanted cells connect with endogenous cells, not just integrate amongst other photoreceptors, but connect to bipolar cells, form functional connections. How do they communicate, and how might that be different in a diseased retina compared to a healthy retina, or a retina that's undergone some degree of degeneration, either partial or complete loss of photoreceptors? And for example, one of the hypotheses that we're actively pursuing in the lab is might that process also be modulated by the actual genotype? Like, might it actually depend on the type of disease that's actually occurring? Maybe efficiency is not gene agnostic. Maybe the efficiency of cellular integration actually does depend on what disease you are trying to treat. And some diseases may not be as amenable to cell replacement therapy as we would hope. Maybe it's not all just one final common pathway. Because we know some diseases cause degeneration of RPE before they cause degeneration of photoreceptors. They're primarily an RPE disorder. There are others that are primarily photoreceptor disorders, ciliopathies and other types of things. So I wonder if that's been a bit of a hindrance. I mean, a lot of the clinical trials that have been tried so far in stem cell therapies have just accepted patients regardless of what gene they have as enrollment. So it's quite a heterogeneous population, right? It's patients who've had degeneration for any cause, and they're not seeing the efficacy signals they'd hope to see. And it's a lofty goal to be genotype agnostic, but maybe we could be genotype specific, or at least create a list of priority genotypes that would actually be amenable to cell transplantation. That's a future-looking hypothesis, but you did ask what might be holding us back a bit. I think a lot has to do with our understanding. If we could understand better about how a cell makes a functional connection, maybe we'd be able to coax these cells to do what we want them to do. But stem cell therapy has always had a higher hurdle to jump, because you're not talking about just replacing. I heard a great analogy from David Gamm recently, and we were at a meeting together, talking on a panel together, and he said, gene therapy is like you're in the house and your furnace breaks down, and the heat goes off in the house, so you got to go in and replace the furnace, but you don't have to tear down the house, right? The house is still there. But in cell therapy, the challenge is your whole house is falling apart.
There's no house.
You got to replace the whole house, and when you replace a house, you got to put in a furnace, right? You got to put in maybe an AC unit. I don't know how luxe you want your house to be, but maybe you got to put in a water heater. You got to put in some appliances. There's a lot that comes with it. And cell therapy's always had a bigger hurdle, right? And you got to find contractors and zoning rights, and you got to find a place to build the house. Yeah, so it's
true. It's a
good
example.
Analogy. You could keep extending it. It's a crazy one.
Yeah, it's a good example. So sounds like if you were going to pick what's more promising in the next decade, for example, you would pick gene therapy, different approaches.
I think we're going to see options. I think we're going to see new pharmacotherapies, like I mentioned for the Stargardt, but I think we are going to see new gene therapies. I touched on only a couple. There's several other trials that are in phase 2 and 3, and I think we're going to see some new gene therapies.
Awesome.
I think one of the things that we saw with RB65 was we were replacing an enzyme. So we needed to replace enzymatic activity. Now we're replacing proteins that have other functions in supporting the primary cilia or structure of photoreceptors, structural proteins. So, there's a lot of work in X-linked retinoschisis, for example, and we're going to see a phase 3 study, the Lighthouse study for X-linked retinoschisis, a very promising compound from Etsina. So a lot of work going on in structural proteins too.
Wonderful. One last thing, if I can maybe ask your opinion about, how about devices? You mentioned Argus already, which is going to die down, unfortunately. Anything in that regard, anything up in horizon with technology-based devices and solutions?
I mean, so much of my work is in cellular and molecular. I probably am. I'm probably woefully, perhaps I won't be able to point to the most prominent examples in that. And for for your listeners, I'd apologize there. I mean, I am aware of continuing research in new prostheses, implants, and chips with more numerous photodiodes that are packed more densely to be able to better stimulate the retina. But I'm not sure that, I mean, that field is still, having to mature, and I'm not sure we're going to see. I mean, I think a lot of these gene therapy studies are frankly just a little further ahead than those. Those are promising, and they'll be helpful for very specific populations. But they also depend on not just how do you implant and connect this device to the retina, but how do you stimulate it efficiently? Do you have to wear special glasses to actually increase the luminance of light properly? How do you modulate the visual information to simplify it enough for these devices to be able to translate it to a signal that can be interpreted by a ganglion cell matrix?
And make sure it sustains and that doesn't get damaged over the years.
Correct, and it doesn't run out of power. So there's a lot of practical engineering design hurdles there, I think, that it'll need to be addressed for that. It's exciting, but it's and I guess the last thing I would mention to your listeners is natural history data. It's not always the most sexy or exciting area of clinical research, but we have, in inherited retinal disease, we have not traditionally had great natural history data for individual genotypes. We and it's interesting that our reach has sometimes exceeded our grasp. Like, we have tried to develop new gene therapies for specific genotypes without really understanding the natural history of that disorder. So we've posited that we might see changes in visual fields, we might see changes in microperimetry, we might see changes in FST without knowing how those changes actually happen over time in those specific diseases by those specific genotypes. And so large consortium-based studies like we're part of that collect this natural history data from patients with specific types of inherited retinal disease caused by gene X, Y, or Z are giving us the information we need to better design clinical trials, because they're telling us what are the most sensitive things for individual diseases that change over time. Like, should we be paying attention to perimetry? And if so, how should we be measuring it to be able to detect meaningful functional change over the time scale of a clinical trial, which is going to be 1 to 2 years, and because that's ultimately what is going to make a regulatory body sit up and pay attention
and compare that to treated arm, natural
to approve, right? You got to have an efficacy signal, and if you don't have good natural history data and you know how this disease changes over time, how will you really know that your intervention is effective? Planning clinical trials without good natural history data is like sometimes putting the cart before the horse. So there has been a huge amount of work in the last 5 to 10 years on natural history data. We're part of a very large study called UniRare, Universal Rare Disease, which is conducting has a number of different natural history arms for a variety of different genes. And I think that will provide more data that will stimulate more interest from groups to try their new compounds in specific genotypes, because they'll be able to better design clinical trials, and we'll be able to support that. Pharma companies don't necessarily have the legs. They develop the compound, but they're not going to do their own natural history studies, right? To know what's going on. It takes decades, right? It takes years, right? It takes 5, 10, 15 years in IRD to collect this data. We got to do it now. And now that we are able to better do genetic testing and find the genes that cause these disorders, we can actually do it, because we can
Wonderful.
identify, find the patients, collect them, and do the work. So that's
Wonderful.
the last plug I put in. I know we're talking about gene therapy, but natural history data is super important in this field. We've been
No,
doing
we
a
want to
lot
talk
of
about
work.
research. What's important IRD? So
Yeah,
that's
research in natural
res
history, that's new since we last spoke about this, and we're doing a
very
lot of
nice.
that now. Yeah.
Well, Dr. Ballios, you delivered. You know, you exactly gave us what we wanted. You know,
Okay.
and sort of top line, what's going on. You
Okay.
know, bring us up to speed, and you know, I really appreciate it. Really, I learned a lot personally, and I'm sure people also learned a lot. And thank you for leading the way. You're one of the world's best IRD specialists, one of the most talented ones. I've known you for many, many years. So
It's
I'm
very
really honored
generous.
that you came on the Retina Channel podcast, and you know, it's a privilege to be a colleague. So thank you for being here.
I appreciate it. Dr. Kushan, it's terrific to work with you and to share, and you always send very thoughtful consultations. I like working, and our patients really enjoy the care you provide. So I thank you. And I really appreciate the opportunity to talk to your listeners about some of this, because IRD is still a very, very exciting space, and they will be seeing new therapies come out, and they should remind their patients and provide them with some hope, because there's a lot to be hopeful for.
I think so. I'll promise everyone I'll bring you back again so for next
Okay.
update.
All right. I look
Thanks,
forward.
Brian.
Thank you. Thanks, Kayvon. Have a good night.
Well, folks, this wraps up episode 98 of the Retina Channel podcast. I hope you enjoyed it and you learned from it as I did. And it's amazing to see how much things have evolved in the field of inherited retinal diseases since we first spoke with Dr. Ballios back in 2017. It was a fantastic overview of where things are today, and more importantly, where things are heading for the future. Thanks again, Brian, and thank you everyone for listening to Retina Channel podcast, and we'll see you on the next episode.